Hi Hires, we are looking at the key area 5 structure of the genome in DNA and the genome unit. Okay, so this key area, there isn't a lot of content statements to it, but at the same time there is a lot to it in terms of what you're trying to say. What we're looking at here is the bringing together of every single thing about the genome. And the genome is every single bit of coding information you have, every bit of hereditary information that is being passed in an organism. Okay, and the genomics is the study of that, of every gene, about where you find it, what its sequence is, what it does, how it's expressed, and how it evolved, and where it might be evolving to. I mean, all of that is massive. It's a huge branch of biology, and there's a lot of detail to it. You're looking to have a kind of overview idea of what the genome is, and then what some of the problems are when we're trying to study it. So the human genome, I've mentioned this before in I think a couple of other places, the Human Genome Project, original sequencing of the complete genome. 2003 managed that. Over 3 billion base pairs, that's quite a lot, and we now think around about 25,000 genes, which is a lot smaller than we thought, but then that codes for an awful lot of proteins. If you go back to the section on proteins and how they're produced, the idea of alternative splicing giving us one gene, many proteins. So actual coding DNA though, that 25,000 genes, is only about 2% of the genome. So genomics, actually studying the genome, is not just about studying coding DNA. It's about studying coding DNA and all of the rest of it around the outside. So this is what we know about the 2% coding. Unfortunately, there is a big chunk in the middle there we can see with the nice big orange blob. About a quarter of it we still don't know what they do. So a quarter of 2% we don't know. But there's some interesting ones that are in here, like we have ligases, which you only know one, DNA ligase that joins things, but actually there's 260 of those have been identified. So these are things that join things together. We've got a whole pile of viral proteins. Well, not a pile, but 7 of them. That's strange in some ways that we've still got those, but we do. This one, we're going to have a little look at transcription factors. Over 2,000 of those have been identified. So transcription factors are to do with helping transcription happen. So some of our coding is to make proteins that then help to make proteins. Okay. Not surprisingly, we've got a pile of receptors up here. We've got over 1,000 of those. There's a whole pile that we know. There are also big chunk we don't. Okay, and then we also have this huge chunk, which we don't really, the 98%, so the non-coding section of our genome. So we do know some of them are protecting the ends of the chromosomes. We have a problem with our DNA in that it's linear. So we've got ends that are just basically available for degradation. There is a lot of sense to having a circular chromosome because there is no end that's exposed, but we have linear chromosomes. So we know that some of the sequences help to regulate gene expression. I'll come back to those transcription factor ideas. We also know that there's some that are transcribed, but we don't translate. So we wouldn't necessarily say that's a gene because we're not making a protein, but at the same time we're saying that is actually a recognized sequence that does something, we just don't change it to amino acid code. So something like tRNA, the sequence for the tRNA, we obviously still need to copy that sequence into RNA, fold it, there's a wee tRNA molecule, but it's not actually translated. So the end caps ones are called telomeres. Now you don't need to know all of this detail, but you do need to know examples of how these known coding could be used. This is a nice example. Okay, what you have are sequences at the end of chromosomes that just stop them being damaged at the end. But every time that you copy that chromosome, you basically lop one of these off at the end. So how many telomeres you have is a really good indicator of how old that particular cell is, how often that cell has replicated. And so overaction of, or underaction more importantly, I would suppose, of the telomerase, which is the enzyme that makes telomeres, is linked to cell aging. And potentially could be linked to trying to prevent cell aging. it's an interesting one. So your other one that we know for sure is that it's to do with how much expression you have. And you already know a couple of these, like you know that we've got a promoter sequence that needs to be locked onto by RNA polymerase. So if you want that to happen, then you need to have that coding section preceded by a non-coding bit, which is the promoter. Okay, there are also things called transcription factors, which can be proteins and other bits that have been translated and then are being used to bind on to bits of the DNA. And we have other sequences that are activators and also preventing transcription. They would be repressors. So giving you a picture of how complicated this might look. Okay, remember that bubble that we drew ages ago when we were doing RNA polymerase? Here's your RNA polymerase here locking over the promoter. But what you've also got is all of these proteins which are all acting together to get RNA polymerase to function. And the only way they function is if they have been attached to these sequences over here. But these sequences, if you look at the DNA strand, is all the way around here. So the only way that these can actually come into contact is if you've got this protein here, which is bending it. So this bending protein, this DNA bending protein, is absolutely essential to get these control elements into the right place for them to hit the activators, which then bind to RNA polymerase, which means that when it's attached to the promoter, it can produce gene A. A lot more complicated than what we've been saying before. Okay, again, you don't need to know the detail, but the idea is essential, that you have other things that are happening in the non-coding section to allow code to be produced. And finally, we have transcribed but not translated. So I mentioned tRNA already, but rRNA you should also recognise as that's not actually translated. That is an RNA form that is just associated with proteins to make the ribosome. And there are other forms of RNA, some of them just found in particular organisms. You don't need to know the examples of them, just know that it's not just the 3 forms of RNA that we have, there are more. So what about all the rest? Unfortunately, we just don't know. So originally that 98%, the stuff that wasn't coding, it was called junk DNA, which it cannot be that, because you wouldn't maintain it in the system otherwise. So it has to be doing stuff. And the examples of transcribed but not translated, your telomere repeats, your expression being coordinated, they absolutely are true, but we just don't know everything. So is it to do with folding of the DNA? Probably. You know, is it to do with the aging of the DNA? Is it more nuanced controls of expression? All of that is possible, but we just don't know exactly. You need to be able to give a couple examples of what we do know it does, so go with one of the other examples of that.